So, uh, we are going to see the chapter which is molecular basis of inheritance. Okay, so in this chapter, mainly we'll be seeing about the genes which play an important role in the human body as well as the other organisms as well. So, first we'll be dealing with all the terms which are very important. So, starting from the first term, which is the inheritance. So, basically, what is inheritance? Okay, simple words, if I mean to say what it is, it's nothing but the process by which you get the characters from your parents. Okay, so it's basically whatever the parental characteristics are there, they are being passed on towards their offsprings. Okay, so this is basically called as what? Inheritance. So the process of passage of that particular genes from the parents to the offsprings is called as what? Inheritance. But related to this only, there is one more thing which is called as heredity. Okay, so heredity means what? It is the process of passing down the genetic traits from parents to offsprings. See, now there is just a minute difference between inheritance and heredity. Inheritance is nothing but what? The process where you will get certain characteristics. Okay, but heredity means what? Genetic traits. Now, genetic traits means what? Some people, they may be having certain kinds of maybe diseases, okay, or certain genes will be altered. So there may be a possibility that the entire family history, if you take, they will have the same things passed on to their offsprings. Okay, so this thing is called as what? As a heredity factor. So characters basically are what? Like the color of the hair, the color of the eyes, okay, the height, all those stuff. Those things are called as what? Inheritance. Whereas genetically, whatever things are being passed on towards, like from the generations, then they are called as what? The heredity. Okay, so but basically, what happens is sometimes there are chances that their characters may be different. So when these characters are different, then we say it's a variation. For example, if you take parents, okay, they are having both are having black eyes, but the child which is born to them, they are having maybe blue or green eyes. So this is what? A variation, because in their family earlier they never had a child with the blue or green eyes, but now it's born. So those things, that thing is called as what? The variation, that is the degree of difference in characters between the parent and the offspring, that is called as what? The variation. Okay, so these three terms are very important whenever you are studying genetics. Okay, so genetics means what? Basically a science which includes the inheritance and variations. Okay, that is nothing but the characters which are being passed from the parents to the offspring. So this study of genetics was first time done by the person called as G.J. Mendel. That is Gregor Johann Mendel. Okay, so he is basically the father of genetics. So he usually started this thing by experimenting on a pea plant. Okay, that is the Pisum sativum, because pea plants have many different characteristics by which he can easily use that plant for different types of experiment, which we are going to see further as well. Okay, so as I told you, whatever the genes or the factors are there, they are being passed from the generation to generation or the parents to offsprings. So what passes actually? Okay, so the thing which passes from this parents to offspring is called as the chromosomes. Okay, so that particular chromosome will be having genes present inside them. Now, as you know that the XX, okay, XY, these are the two types, okay, which mix together and it either gives a male or a female child. So, in this chromosomes, genes will be present, and whatever variations or similarities we have with our parents, that is due to what? The alterations in the genes, which are generally present where? In the chromosomes. Okay, so usually whenever the XX and XY chromosomes combine together, okay, the haploid plus haploid, they will be, which combines to form what? Diploid. So hence the chromosome which that baby will be having will be in diploid manner. And that particular thing is called as what? Allele. Okay, so that particular gene which is present, okay, in an offspring, that is called as what? Allele. Fine. So how do you make out that it's a physical characteristic change or internally if there is a change? Okay. So there are two basic terms which are used over here. One which is called as the genotype and the other one is called as what? The phenotype. Okay. So I'll just mark this. Yeah. Fine. So genotype and phenotype. Okay, now simple difference between genotype and phenotype. Phenotype is nothing but the external appearance, whatever you can see and recognize the difference. Like for example, the height, okay, the color, whatever things are there, they are all what? The phenotype means external morphological appearance of that expression genes is called as what? Phenotype. So genotype will be what? Internally, because internally by seeing that particular organism you cannot say what changes are there, hence genetically the changes may happen, so hence they are called as what? The genotype. Okay, so there are many different characteristics which we will be seeing now. Fine. So just a second. Yeah. So next is the Mendel's law of inheritance. Now, I already told you this Gregor Mendel, he put forward an experiment on pea plant, which is generally known as what? Pisum sativum in scientific name. Okay, so he used many varieties and combined together to see what actually brings out. Okay, so this thing was done or he studied this genetics in a span of 7 years. Okay, so he used two techniques of pollination, as we all know, that is the self pollination and cross pollination. Okay, so to get a pure breed, he used the self-pollination, means there were no kind of combinations. That's why it's called as self-pollination. Whereas cross-pollination means whatever desired characteristics is that you can incorporate into that particular thing. So hence they are called as what? Cross-pollination. So whenever you do, what do you say? Self-pollination. Okay, so in the self-pollination, whatever you will get is a true breed. So there will be no kind of combinations or hybrid thing in that. But when you do a cross pollination, you are going to get what? The characteristics which you need, and they will be contrasting instead of the similar characteristics. Okay, so to do this experiment, he took 7 pairs of what? Contrasting characters, which produced 14 breeding pea plant varieties. So let's see one by one what are those varieties. Okay, so these are the characteristics, okay, which he used to study. So he used seed, flower, pod, stem, okay, and he took different characteristics such as the color, the height, okay, how it is, the size, and all those stuff. He took like that 7 contrasting characteristics and he combined them together to form certain things. Okay, so now whatever the 7 characteristics he took. Now, as I told you, genetics combines the inheritance as well as the variations, right? So first we will be seeing what? The inheritance of one gene over here. Because when you're seeing the 7 characteristics at a time, it may be a little confusing. So one by one, we'll be seeing all the things, right? So inheritance of one gene. Now, if you see the now he took the basic 2 characteristics. Okay, that is one is the so he took here 2 characters. Okay, so one is the tall and another one is the dwarf. Tall and dwarf. Okay, so one is the dominant character because it will express always, and the other one is the recessive characters, means it will be suppressed to some extent until and unless the dominancy is broken out. So inheritance of one gene means what? He took in this two things. That is, the height of the plant basically was the character he opted for, and that was tall and dwarf. So he collected those seeds which were tall and dwarf, and he sowed them and grew those particular plants. So whatever he got in the first set, okay, so that is called as what? First filial progeny, or F1 generation. Okay, so when he crossed the two characters, tall and the dwarf, whatever he got in the first generation, it is termed as what? First filial progeny, or the F1 generation. Okay, so now whatever products he got from this F1 generation, he again recrossed the thing. Okay, so from the F1 generation, whatever result he got, he again crossed it, means F1 into F1 he did. So he got F2, which is called as the second filial generation or second filial progeny. So when he did this two F1 and F2 generations, whatever he got was what? Contrasting characters. Okay. So what were the results, okay, after crossing? So if you see over here in the first thing, the first result was what? In the F1 generation, when he crossed the tall and dwarf, he got what? One dominant. Okay, so whatever the progeny or whatever offsprings he got was a dominant one. Okay, so it always resembles what? One of their parent will be dominant. That was the statement he got from crossing, or the product which he obtained from what? The F1 generation. But then when he recrossed the F1 into F1 to get the F2 generation, that represented a mix or a hybrid that was of the dominant as well as the recessive characteristics. And how much was the amount is the 3/4. Okay, so 3/4 was dominant, whereas the 1/4 was recessive. Okay, so typically when you take the product in the ratio format for the F1 generation, the like if you cross any characteristic, okay, it's not like you have to cross the tall and dwarf only. If you're taking the color or size or whatever things, it is shape, okay, whenever you cross the whatever F1 generation you're going to get will be same. That is, it will be 1:2:1 for the what? The ratio genotypically, whereas for the phenotypic ratio is 3:1. Now, phenotypic, genotypic, you already know. Phenotypic is the morphological character, whereas genotypic is a genetic character. So this will be same. But when you take the ratio of the F2 generation, then it will be a very big ratio. So it is not possible to write all the ratios together when you cross many characters together. So hence, this is the basic format which will not change for the F1 generation ratios. That is 1:2:1 for the genotypic ratio and 3:1 for the phenotypic ratio. So if you see over here, in both the generation, there was some kind of resemblance of that particular offspring with the parent. Right? So when you blend this different kinds of characteristics together, okay, you will get what? A new character in that particular progeny of the offspring. Right? Now here, whenever the crossing is done, it is done in two ways. So either it is a monohybrid cross or a dihybrid cross. Okay, so first let's see what is a monohybrid and dihybrid. So okay, so if you see over here, okay, that the TT means tall and small tt means dwarf. So if it's a capital T and small t also, they are considered as tall only. So only if two T's are small, then only you can say it's a dwarf plant. Okay, means it's a recessive characteristics. So monohybrid and dihybrid cross, the main difference is what? Whenever you cross by taking only one selected character, you say it's a monohybrid cross. Okay, so one selected characters means what? You have a homozygous tall and a homozygous dwarf. So homozygous and heterozygous are the two terms. Okay, so homozygous means what? Both the characters or both the letters will be same. For example, in the first one, if you see capital TT, whereas in the second one, it's small tt. So both are similar. So hence it's called as what? Homozygous. But the two things over here down, if you see after crossing whatever you got over here, it is what? One capital T and one small t. So they are called as what? Heterozygous characteristics. Okay, so this is the what? The monohybrid cross. So only one selected characteristic you're going to take for a monohybrid cross, whereas for a dihybrid cross, you will be taking two contrasting characteristics for the crossing purpose. Okay, so you can see here clearly how after the crossing, you're getting the F1 generation as well as the F2 generation. So already I told you the ratios, that is genotypically it's 1:2:1. So how can you make out that it is 1:2:1? Now, if you see over here in the last, okay, there is just one capital TT and one small tt. Whereas in between the two which you're having is a hybrid, that is one capital T and one small t, right? So if you count this, okay, so one capital TT, so you put here 1, then 2 small tt, two hybrid ones, so you put here is to 2, and then 1 small tt, so you put is to 1. So this is basically 1:2:1. This is genotypic ratio. But external, if you want to read this characteristics, we have 3 tall and 1 dwarf. So the phenotypic ratio becomes tall:1 is equal to 3:1. Okay, so whatever characteristics you are taking, this is what you are going to get. Okay, so yeah, so based on this, okay, monohybrid cross, that is the cross which is made only by taking a one contrasting character. So here he made 3 laws. Okay, so he proposed 2 out of the 3 laws, which was related to the inheritance one, that is the characteristic one. So now what were the 2 laws? So first law is called as the law of dominance and the second one is called as law of segregation. So now what is law of dominance and law of segregation? Dominance. Okay, dominance means what? One such character will be present in that particular gene which is always dominating. It will only express making other cells suppressed. So this is called as what? Law of dominance. So what does it state? It states that among the two contrasting characters, the dominant one will prevail in the offspring, means it is going to express in that particular offspring. So it explains why only one character was expressed in what? The F1 generation. Because if you see in this, only one character you can see. That is which character? Tall, right? You don't see any dwarf over here. So this is a tall characteristic which has been expressed in the F1 generation. So that's called as the law of dominance. Okay. So hence you got the generation as 3:1. Now law of segregation means what? Okay. So whenever a gamete is being formed, so only one allele. Now what is allele? Allele is a character of that which is present in that particular gene. So only one allele from the pair of chromosome will get transferred from each parent. Okay, so that is called as law of segregation. If you take two parents' chromosome, from that only one character will get transferred to that offspring from each of the parent. Hence, whatever genes or gametes you're going to get will be in the pure form. Okay, so hence this law of segregation is also called as law of purity of gametes. Okay, and it's also basically the universal law. So law of dominance means it will only express and suppress the other characteristic in the offspring, whereas law of segregation means what? Here, whatever the when the gamete is being formed, only one character or one allele from the pair of chromosome will get transferred from each parent. That's called as what? Law of segregation. Okay, but it is not always that whatever the dominance is there, it will be a complete dominance. Okay, so sometimes it may also be incomplete. So you have one more law related to the incomplete dominance, which is called as law of incomplete dominance. Now, what is this law of incomplete dominance? Now, complete dominance means what? That dominant character will surely express no matter whatever happens. So 100% surety also you can give. But law of incomplete dominance means what? Now, there will be no dominant or recessive gene which will be present. So hence, what is written over here? Both the parent will be expressed in the offspring, but in an intermediate or mixed manner. Means you're going to get a combination of the tall and the recessive one. So here, the two examples which they have taken for the combination is the snapdragon and the dog flower. Okay, so one will be having the red, another one will be having the white. So here, if you take these two plants, the red flower will be dominant over the white. Okay, when it is what? In the homozygous condition. But when it is expressed as an heterozygous condition, you get two things, that is dominant and the recessive. Now, already I showed you in case of the tall and dwarf, how in the first generation you're getting the heterozygous gametes, which you get only tall plants. But when you just recross it again, whatever you're getting is what? A mixed or a hybrid one. So the same thing is explained over here. In short, incomplete dominance means neither dominant nor recessive gene will be present. You will get an intermediate results over there, and mostly the condition will be heterozygous. So as you can see over here, in the genotypic ratio, okay, that is 1 RR. This they have taken over here is the what do you say, how the seeds appear, whether it's round, wrinkled, red color, white color, those characteristics they have taken over here. Okay, so you have the different genotypic as well as the phenotypic ratio. So what happens over here is, in an incomplete dominance, whatever the phenotypic ratio, which is generally I told you is 3:1, that changes over here to 1:2:1. Okay, so generally if they ask you what is the phenotypic ratio, then you have to say it's 3:1. But if they give a condition that it is what is it? It's an incomplete dominance, you say that the phenotypic ratio will be 1:2:1. Okay, so this is all about the incomplete dominance. In the same way, you have one more dominant type, which is called as codominance, which you generally see in the blood groups. Okay, so okay, so codominance. Now, co means what? Together. Okay, so codominance means what? Here, there will be no dominant gene or recessive gene. Okay, so whatever genes means from the parents, both the parents will be there. They will act dominant and they will express themselves simultaneously as a heterozygous gene. For example, now I'll tell you how it happens. Okay, so here they have taken the ABO blood group, which is general blood group of the human beings. So here you have one gene which controls this ABO, which is called as the I gene. Okay, so here what happens is three types of alleles you have. Now, as you know, A, B, and O, these are the three types of blood group. So either the person will be A, B, AB, or O. Correct? So one is the universal donor and the other one is the universal acceptor. Correct? So here if you take A, B, and O, or AB, whatever things you're going to take, here what will happen is one will be dominant, okay, whereas one will be recessive. For example, that particular I factor which is there. Okay, so here if you take this A, in this A, what is the genotype which we are getting? Now A is the phenotype, okay, whereas the genotypes which we got is AAAI. Okay, so three dominant and one recessive we have. Okay, the same is repeated for what? B, AB, as well as O. So when it comes to O, both will be recessive, small ii. Whereas for the AB, you have capital II, that is IAIB. Okay, and respectively, whatever the genotypes are there that you have to make out. So the genes which control the blood groups are called as what? The I genes. Okay, so in a codominance, neither dominant expresses itself, neither recessive expresses itself, but when they are expressing together, that is simultaneously, they will give you an heterozygous gene. So hence, this blood groups are being formed, that is AB and O. O is the universal donor and AB is a universal acceptor, right? So this is all about what? The crosses and what happens when you cross different similar heterozygous condition as well as the homozygous condition. Okay, now how do we actually cross this thing and we get a particular outcome? So for this, there is a method which is called as what? A Punnett square method. So a Punnett square method is nothing but a square box you will be having, which will be divided into 4 or 8 boxes, respectively, depending upon how many characters you're going to combine, okay, to get that particular result. Okay, so R.C. Punnett was the person who developed this graphical representation so that you can calculate more easily the genotypes of an offspring. Okay, and the example which they have taken over here is the tall and the dwarf. So whenever you're doing a Punnett square, you basically have two types of crosses. Okay, that is the back cross and a test cross. I'll just draw it, just a second. So the back cross, okay, so here's the, yeah. So suppose if you're doing a back cross, okay, so I'll take the example of the same thing, the TT and small TT. Okay, that is the homozygous tall and the dwarf. When I combine together, what I'm going to get is a capital T and a small t. So basically, whatever you're getting over here is a heterozygous tall. And again, you recombine with the capital and small t, that is the heterozygous format. Now over here, what you're going to get is a 50-50 percentage. Okay, so 50-50 capital TT and capital T, small t. Okay, and on the other hand, what you will be getting is the capital T, small t and small t, small t. These are the 4 ratios which you will be getting when you are doing a back cross. Okay, so the back cross is what? It's basically a cross in which the offsprings from F1 generation is crossed with either one of its parents. So very important point over here is you're not just test crossing the product which you got in the F1. Instead, whatever product you have got over here in the F1, you're crossing with any of the one parent. Okay, not self cross. So that is the difference over here. That's why it's called as what? Back cross, and whatever you get in the F2, it will be a 50-50 percentage. Okay, so that's why I have written 4 products over here. So here I have generally crossed the this product. Now here you will get is a capital T and a small t, and I have just crossed it with what? The small tt. So this is the products which I am going to get. Okay, so this is called as what? A back cross. A cross which is done with whatever product you are getting from in the F1, you are crossing it with either of one parent. So that is called as what? The back cross. Now moving towards the second cross, which is the test cross. So test cross. Okay. So test cross means what? Here, there will be the crossing of F1 progeny with only a recessive parent. Okay. So hence it's called as what? The test cross. The same thing. Okay. I'm going to just draw over here. That is, if you take a TT into small tt, whatever you get is a capital T and small t, which is a heterozygous. So when you cross it, that is when you test crosses. Now when you draw a Punnett square, now generally over here, two characteristics, you just have to put it here. Okay, so it will be like this. So here in the F2 generation, what you will be getting? 50% of the capital T and small t and 50% of the small t. So this is called as test cross. Okay, so in back cross, you may take any of the parent. Okay, it's not that you have to take recessive only, or you have to take the dominant parent only. But when you come to a test cross, the condition is what? Whatever the F1 progeny you're getting, you just have to cross it with the recessive parent only. Okay, so that will determine the unknown genotype of the individual. So this is the two types of crosses, that is the back cross and the test cross. Now, whatever we have seen over till here is the inheritance of just one gene. So if you're using that one particular gene, what are the things you may get, that we have seen over here. Okay, now in this two thing only, the whatever the two laws I told you, that is the dominance and the law of segregation, these are applicable till here. Okay, so whereas the third law which is there, that is the law of independent assortment, that we will be seeing now further. Okay, so how, like one gene inheritance we saw, in the same way there is one more thing which is called as inheritance of two genes. Okay, so the basic difference is that in the two genes, whatever the genotypic phenotypic ratio will be getting, it will be a little more longer as compared to inheritance of one gene. Okay, so that means you're going to take total what? 4 things to cross. So you're going to get, instead of this 4 boxes Punnett square, you will be getting 8 to 16 boxes Punnett square over there. Fine. So just so inheritance of two genes. So inheritance of two genes means what? He crossed different characteristics, that which is homozygous round and yellow pea with the homozygous wrinkled and green pea. So whatever he took over here for the crossing, what was the homozygous plant. So here if you see, okay, now this is how it appears to be. So you have the parent, okay, which consists of the round and yellow in the first box and the green and wrinkled in the second box. So whatever F1 generation you're going to get over here is what you can see. How did the F1 generation, what you got over here is a heterozygous products you got over here. That is, you are going to use those products for what? The generation of the F2 thing, right? So now here, instead of 1:2:1 and 3:1, whatever the phenotypic genotypic ratio is there, it is more, as you see over here. The phenotypic ratio itself is what? 9:3:3:1. But when you go to a genotypic ratio, it will be more big. That is 1:2:1:2:4:2:1:2:1. That's why I told you, whenever a monohybrid cross is made, phenotypic genotypic ratios, whatever you're getting, will be in a simpler format. You can easily count it. When you go to the dihybrid cross, means when you are making a cross between maybe you can say 2 or more characteristics, then whatever products you're going to get will be more. So if from that phenotypic ratio, still you can make out. If you see in this Punnett square, external characteristics still you can count and make out. But when you come to the genotypic ratio, it is a little complicated part. Okay, so if you see, the ratio is very big. Okay, so here, because they have used the two contrasting characteristic, which type of cross it is called as? Dihybrid cross. So in the first two, we saw the monohybrid cross, which in which we saw the law of dominance and law of segregation. Whereas in the second, that is in the dihybrid cross, you have the third law, basically, that is the law of independent assortment. Okay, so what is law of independent assortment? It is nothing but the combination of two pairs of traits in a hybrid segregation of one pair of character is independent of the other pair of character. Means even though if you're combining more contrasting traits together to bring it into a hybrid format, still what happens is one pair of character is going to separate out and going to express independently, leaving the others. This is called as what? Law of independent assortment, which is followed by the dihybrid cross. So